Hello, and welcome to Chapter 6. We are going to be talking about DNA and gene expression through this chapter. The first thing I want you to do is to think about what you think about when you hear the term DNA. What do you think of? Do you potentially think of the shape of DNA looking like a double helix? We'll look at that later if you're not sure what I'm talking about. Or maybe you think of genetic coding or something that makes you you. Or maybe you think of how your parents look and then also how you look. There are a lot of things that you probably think of when you hear that term DNA. The technology surrounding DNA has greatly advanced in the last roughly 50 years. It's a very powerful tool for identification of individuals, especially when we talk about maybe the criminal justice system. But there's also things surrounding DNA technology, like modifications to DNA, and eliminating diseases, creating organisms that were previously extinct, potentially. You can think very Jurassic Park-esque. There's a handful of examples that we'll talk about with biotechnology in the next chapter, that surround a lot of that DNA technology. However, for this chapter, it is just kind of a precursor to we now modify DNA. We can also look at DNA very closely, but how did we get here? There are three individuals who were very instrumental in figuring out what the DNA molecule looked like and also what it contained. So we have two gentlemen, James Watson and Francis Crick, who were able to figure out the exact structure of DNA. In fact, Francis Crick's wife, Odile, was the individual who sketched that first rendering of a double helix DNA structure. We also have Rosalind Franklin to thank, who was integral in understanding the molecular structure of DNA and something that we will talk about here in just a second, RNA. When we look at the structure of DNA, that is going to be composed of three important parts. So if we were to look at DNA, the structure of it, it looks like a ladder that's been twisted. So if we first look at the sides of the ladder, that is going to be composed of alternating deoxyribose, which is a 5-carbon sugar, alternating with phosphate. So that is the outside of the ladder. On the inside of the ladder, the rungs of the ladder, those will be nitrogenous bases: adenine, thymine, guanine, and cytosine. So these three very important parts create what we call DNA. Okay, so from here we are going to talk a little bit about RNA, but we have to kind of compare it to DNA in order to really understand what RNA is. So if DNA, the rungs of the ladder, are made of these base pairs, which they have to follow a pattern. Adenine will pair with thymine, thymine will pair with adenine, cytosine will pair with guanine, and guanine will pair with cytosine. That will be the pattern forever with DNA. Now, DNA, because it has those pairs and two sides of the ladder that are those alternating sugar phosphates, we are going to see that DNA is double-stranded. There are two sides to the ladder. DNA is copied in order to make RNA. Now, it's not going to be both strands that are copied. It is only one side of the ladder that gets copied into RNA. RNA is single-stranded, and instead of having base pairs that are going to have a partner, it's just one, and that's because it's just the copy of one side of the DNA. Now, when we copy DNA into RNA, thymine will copy over into adenine. Adenine, however, will not copy over into thymine. It will copy into a different base called uracil. Cytosine and guanine will still have that same base pair structure and partnering system. So cytosine will pair with guanine and guanine will copy over into cytosine. So a big difference between our DNA and RNA is that DNA will have thymine, RNA will have uracil, DNA is double-stranded, RNA is single-stranded, and is created as a copy of one side of the DNA. Now, why do we care about any of that? Well, our A's, T's, C's, and G's are going to make up our DNA. Now, sections of DNA that code for a specific thing that is a physical structure on your body or on another organism's body is a gene. So genes are sections of DNA. These sections will contain coding with those A's, T's, C's, and G's in a particular order and fashion that are essentially instructions to make those physical structures. Those physical structures are going to be made out of proteins. So pretty much everything that is a physical structure on an organism leads back to those proteins being made from the instructions, which is the genes. All living organisms are going to have DNA that are instruction manuals that lead to those physical structures on the organism itself. I have quite a few definitions that I need you to know from this chapter. In fact, this chapter is very definition heavy. We first have the term genome, which is probably a term that you've heard before. The genome is an organism's complete set of DNA. I would also add into your notes that if we want to kind of have a nice little association of what a genome is, compare it to something else, it's like a complete cookbook. It is the whole book front cover to back cover. Chromosomes are going to be one or more unique pieces of DNA, and in the context of a book, chromosomes would be like a chapter. When we get to the term gene, these are specific sequences of DNA. This would be like a page within a chapter, because there's multiple pages that make up a chapter, so we have multiple genes on our chromosomes. And then the locus is going to be the page number, so it is a position of a gene on the chromosome. So it is that specific page number that page is on within the chapter, within the book. A few more definitions of terms maybe you have not heard, and that is alleles. Alleles are different versions of a gene that code for the same feature. In the example that I have here, we have a gene that leads to the color of the petals of a daisy. So we have different versions of that gene. So it all leads to a color, but because the genes can be slightly different, that leads to a little bit of variety. So for allele 1, that is one version of the gene, it is going to create orange petals. Allele 2 is going to lead to yellow petals because it's a different version of the same gene. And then the last is allele 3, giving us a whole set of variety where we have purple petals, again still coding for the same trait, which is a characteristic or feature of an organism, which is the petal color. But we have different alleles that lead to that different variation of the same trait, which is petal color. Now, not all DNA is going to contain instructions for making proteins. There is no relationship between genome size and genome complexity. In fact, humans have a decently small genome size. However, we're decently complex compared to something like an amoeba, which is not the most complex organism, but they do have a particularly large genome size. A lot of DNA in various organisms is actually non-coding. I like to think of it as filler DNA. It's those A's, T's, C's, and G's, except they're not part of a gene. They don't lead to a physical structure or a trait or a protein. What we can kind of commonly call this is junk DNA. So this non-coding DNA is made up of essentially repeats and fragments, or sometimes we call this a pseudogene, so a fake gene. Areas within actual genes that are non-coding, which it can exist, are called introns. So these are just areas within a gene that don't actually code for anything. Okay, a few more definitions, and I would highly recommend that you memorize these two terms because they will come up frequently in the next couple chapters as well as in lab. Genotype is going to be the genes that an organism carries for a particular trait. A genotype is made up of two alleles. You're going to have two sets of essentially every allele or in every gene. Now, you inherit one set of DNA from your mom, and you inherit the other set from your dad. You have a maternal and paternal set of DNA that combines to make your full set. So you have 23 chromosomes from your mom, 23 from your dad, giving you a total of 46. There are pairs of chromosomes, which leads to pairs of genes. These pairs of genes are going to be your alleles, right? These pieces of genetic information are going to code for a particular trait. Now, when we talk about the product of a genotype, that is the phenotype. That is the physical manifestation of the genes for a particular trait. So an example we can think of is that your parents passed down genetic information to you for your eye color, hair color, skin color, whether or not you have a pointy or round nose, whether or not you have flatter or not so flat or rounded fingernails, whether or not you can curl your tongue like a clover leaf, all kinds of things. All those physical structures are inherited genetic information from your parents. The actual physical trait, so eye color, hair color, skin color, nose shape, whether or not you can curl your tongue, that is the phenotype. That is the physical manifestation of your genes that you inherited from your parents. Okay, so this is kind of the big picture as far as gene expression goes. So sometimes called the central dogma of biology, this is what we're going to kind of focus on for the next little bit of this lecture. So the big picture here is that we start with DNA, and that is going to be our genotype. So that is going to be those two alleles together making that gene, right? So DNA is going to go through a process called transcription. Now, I'd focus on transcription in the next couple of slides. Just know that transcription is a process that is going to copy the gene in DNA to make a middleman, which is RNA, that single-stranded piece of information. When we convert DNA into RNA, we are then able to do another process, which is translation, because we can't go straight from DNA to a protein. We have to have the RNA. And translation is going to be quite literally like what you think of with the word translation, like going from one language to the next. RNA is going to be our A's, U's, C's, and G's that are going to be translated into amino acids. Amino acids are then going to build on each other to create a phenotype made up of proteins, so that physical structure. So we're going from DNA to a physical display. And I have another term for you on the next slide. You can also think of it as going from a gene to a protein. Now, I do have some fabulous descriptions of transcription and translation up here. I'd make sure you know that. But we are going to go through the whole process of going from a gene to a protein. So how this works, how do we start with DNA and we get that physical structure? So if we look at the DNA strand, so potentially looking at a specific chromosome, on that chromosome we are going to find genes. We are going to take one of the genes, we are going to unzip it or unwind it, because remember there's two sides to DNA, it's double-stranded, And we are going to run an enzyme called RNA polymerase along that unwound gene. That RNA polymerase is going to produce that copy of DNA, which is going to be single-stranded, and we drop the thymine, and instead we use uracil, and that is that RNA strand. Now, it's important to know that in eukaryotes, this is happening in the nucleus. That is that purple area that we can see in the illustration. So, up to this point, we have unwound the DNA, made a copy of one side of the DNA, and we are producing an RNA strand. That is transcription. From here, the RNA is going to leave the nucleus and go into the cytoplasm, where it's going to run into a ribosome. Now, if you remember from earlier chapters, I told you you should always associate ribosomes with protein, and this makes sense because by running the RNA through a ribosome, we are going to read the RNA 3 bases at a time, called a codon. We'll get to that, don't worry. Called a codon, and that is going to build the amino acid chain. Amino acids are the building blocks of proteins. The ribosome is doing the synthesizing of the protein, so the creation of the protein by building amino acids as it reads the RNA sequence, until you get to the end where you have that finished product, that protein molecule. Now, if that didn't quite make sense, you can do one of 2 things. One, just keep going, and I'm going to go over it again, and then we're going to go over it again and again. Or pause here and go back to the beginning of this slide. Okay, we are going to break it down section by section. So the first part is transcription. This is where the information that is coded in the DNA is going to be copied into RNA. How this occurs is the first step is the RNA polymerase is going to recognize and bind to the DNA where the gene is starting. It is then going to move down one strand of the DNA, so only one side, and it is going to transcribe the RNA, transcribe the DNA into RNA. Now, I would focus on the C in transcription. I want you to associate the C in transcription as copy. We are making a copy of the DNA. From there, the RNA is going to run into what is essentially a termination site or a stop sequence in the DNA that is going to lead to the RNA polymerase essentially stopping making the RNA. From there, it's going to cap and process, and essentially what this means is the DNA is going to rewind and that RNA is going to be kind of finished. Once again, when you think of translation, I want you to think of converting a language into another language. In this case, we are converting the A's, U's, C's, and G's into amino acids. This is kind of the important part. So as the RNA is being read, just like you would with a word, the words, quote unquote, that we are reading on the RNA are called codons. Codons are 3 base sequences on the RNA that match with a part of our ribosome. Don't worry about the kind of specifics of that. Just know that it's matching a part of the ribosome that is going to produce the amino acid that is specific to that codon. And I'll explain that here on the next slide. There are 20 amino acids. You don't have to know all of them. The only one I want you to know is the starting amino acid or starting codon, and that is methionine. Methionine is going to be the codon AUG. That is the only one I want you to know. But I do want to point out that on this key, which is a codon table, it is showing us all the different combinations that you can get with A, U, C, and G. And again, we are only reading these 3 three bases at a time to produce a specific amino acid. You're going to notice that for a few of these amino acids, there are multiple combinations for the codons that lead to the same amino acid. This is actually a safety feature when it comes to our DNA, that if the DNA makes a little bit of a mistake, and don't worry about this for now, we'll talk about it more when we talk about DNA replication. But just as like a little heads up, it is a safety feature that if our DNA, as it replicates, makes a mistake, instead of CAU, it puts CAC, it's going to still produce the same exact amino acid, which is histidine. So it's just a safety feature to have multiple codons code for the same amino acid. We will use this codon table in lab. You will not use it in lecture. It's just something I wanted to show you. Now, I'm not going to go too in detail of translation, other than to say make sure that you know that there is a start sequence and a stop sequence that indicates on the RNA when the protein has begun and where the protein stops. Now remember, we are building the protein through adding amino acids as the RNA is read three bases at a time, called a codon. Now, again, you are building or assembling the protein with this chain of amino acids. That's kind of the important part. Okay, from here, I have 6 easy steps that I want you to memorize the order of for transcription and translation. The first step is DNA is unwound by RNA polymerase. The second step is that RNA polymerase will make a copy of just one side of the DNA, and this is going to make a single strand of RNA. The third step is the RNA strand leaves the nucleus and enters the cytoplasm. The fourth step is that a ribosome, using 3 bases at a time, which is called a codon, will create an amino acid. The amino acids are going to build to create a chain until they hit a stop sequence. And the very last step is that a protein is produced from that chain of amino acids. Once again, I do want you to know all 6 of these steps in order. And I want you to think of it kind of like a story. We are telling a story of how DNA goes through a process to become a physical structure. So as long as you know the general idea of the story, you can put the steps in order, and you will have to do that for the exam. Now, let's pull it all together back into the big picture, and I'm going to do this again, but with a little bit more detail. So we are starting with our double-stranded DNA that is made up of A's, T's, C's, and G's. Your DNA is genetic information that will code for a particular trait. That is called a gene. Now again, our DNA is double-stranded, and on our DNA we find our genotype, that genetic information that codes for a particular trait. From there, our DNA, the A's, T's, C's, and G's, just one strand, is going to be copied from DNA into RNA. That is the process of transcription. From there, the product is RNA. RNA is a copy of genetic information that is single-stranded, and instead of A's, T's, C's, and G's, it's A's, U's, C's, and G's. So remember, DNA has thymine, RNA has uracil. RNA is then going to be translated into a protein. That process is the conversion of RNA into amino acids. The amino acids are going to build on each other to create a protein, and remember, we are reading the RNA, the A's, U's, C's, and G's, 3 bases at a time, called a codon. A codon codes for a specific amino acid. The order of the amino acids is going to determine what type of protein we're producing, and that protein is going to be the physical manifestation of the genotype, the phenotype. So we are going from DNA to RNA to a protein. We are going from a genotype to a phenotype. Okay, so you don't always want your genes to be producing proteins, not consistently or constantly. You do want breaks in between when the genes are active. Now, the whole process of going from a gene to a protein is gene expression. We do have ways to regulate this expression of a gene. So that is going to be whether or not the gene is turned on or turned off, not dissimilar to like a light switch. When it is turned on, that means the protein is going to be produced from the gene. If it is turned off, the protein will not be produced. There are a handful of different mechanisms that lead to this regulation of a gene. We are going to talk about them. Quite a few of them can be regulated by environmental factors, but others are going to be a little bit different. Okay, so once again, cells have kind of a wide variety of mechanisms that control that expression of a gene, whether or not a protein is produced. We call these transcription factors, and in fact, a transcription factor is going to be a protein that binds to a part of the gene that is going to either have a positive or negative control in gene expression. So everything kind of comes back to proteins. Yes, proteins will stop or start the production of another protein. Everything comes back to proteins, guys. So the positive control is going to initiate or speed up gene expression. So that's kind of the on switch. It can also be the accelerate switch. You also have the negative control, which is going to be turning off the gene or slowing down gene expression. And one of the things I want to get across is where we see these transcription factors be really important is during the initial development of an organism. So you can think of like a human fetus. So a fetus is going to need a lot of these transcription factors to turn on and turn off gene expression to start producing those structures that will become potentially something like fingers. So during a particular stage of fetal development, your hands have skin webbing between your fingers. You don't have individual fingers. You have something kind of like a paddle. In order for your fingers to separate, these transcription factors are really important. They turn off gene expression of the proteins that make the skin between your fingers, and those cells will start to disappear. So this is a process called apoptosis. Don't worry about that term for now. We'll talk about it in a few lectures. But just know that it is a process that is going to turn off the gene expression that makes the skin between your fingers, and essentially it separates out your individual little fingies. Okay, another term that I know you've heard of, and that is a mutation. Now, mutations are any alteration to the sequence of A's, T's, C's, and G's in DNA. Now, mutations are commonly misrepresented as always being bad, and they're not. They are not bad things inherently. They are the basis for evolution, and we'll talk about evolution a little bit later, a few chapters from now. But just know, mutations are not always bad things. Not only that, even bad mutations can turn into what is a trait in an organism. Essentially, everything that we see as kind of normal traits in humans probably stem from some sort of mutation from our base species, which again, that is not a bad thing. Now, some mutations are beneficial and some can be detrimental. In humans, mutations that we consider detrimental would be things that are like diseases. So I'm not talking about like a virus or a bacterial infection. Those are not mutations. I'm talking about diseases that are inherited from parent to offspring or potentially developed due to something like a carcinogen. We'll talk about that a little bit later. There are also neutral mutations. These are changes in the DNA sequence, but they have no negative or positive effect on the organism. Sometimes they just exist. They just don't do anything. But it is still called a mutation because it's a change in the base, the sequence of the bases of the DNA. Okay, so we have two types of mutations. The first is mutations that are found in non-sex cells. So something like lung cancer is mutations that occur within an individual, but they can't be passed on to the offspring. So this is typically something that happens due to the presence of a cancer-causing agent. So lung cancer is commonly caused by smoking cigarettes, which are filled to the brim with carcinogens, cancer-causing agents. And that increases the odds of the cells within the lungs to develop this mutation, and thus that is cancer. We'll talk about what cancer is again in a few lectures. A lot of this is kind of like setting down groundwork for a lot of lectures that are to come. Another type of mutation is mutations that can be found in sex cells. Now, typically, sometimes, not always, there are no adverse health effects to the individual who is carrying that mutation. The mutation itself is in the DNA that is going to be in the sperm or egg cells called gametes that then combine to create an offspring. So some individuals are carriers of mutations without knowing that they're carriers. They just don't display the condition. However, there are cases where individuals do have a mutation and they can pass it on to their offspring. An example being dwarfism. Individuals who have dwarfism have a higher likelihood of having offspring that also have dwarfism, and thus that passes on that mutation. Another example would be someone with cystic fibrosis. That is a mutation that they carry, and if their partner also carries that mutation but doesn't display it, there is a higher likelihood that the offspring of those two individuals will also have cystic fibrosis. Now, let's talk about major changes to DNA, so those changes in the A's, T's, C's, and G's that lead to different mutations. We have point mutations and chromosomal aberrations, and we'll talk about the difference between them here on the next slide. Okay, so point mutations are going to be a change in one base. So 1A, 1T, 1C, or 1G. So I do want you to see at the top of this illustration, we have our normal DNA. So our normal A's, T's, C's, and G's in the correct order, or a normal order. That is going to lead to normal RNA, which leads to a normal protein sequence. Because if we change the DNA, it changes the RNA, and the RNA, if it is different, it could change the protein because it changes the order of the amino acids. For our first point mutation, this is a nucleotide substitution, meaning only one of our A's, T's, C's, or G's is going to be swapped out for a different nucleotide, a different base. This is going to change one of the codons in the RNA because the DNA has changed, and that can change one amino acid, which could change the whole protein. This doesn't always happen. So even if substitution does occur, it might not change the protein. It's still considered a mutation. It would just be neutral. If it doesn't change the protein, it's just neutral. In this case, an example of a nucleotide substitution condition found in humans is sickle cell. Sickle cell is where just one base is changed and it changes the whole protein that leads to the structure of your red blood cells. Now, the normal appearance of a red blood cell, it kind of looks like a dented in donut. The purpose of your red blood cells is to carry oxygen. If you have this mutation, it leads to your red blood cells having this crescent moon shape that is the mutation. Now, the problem with it is those crescent moon shaped red blood cells cannot carry oxygen the way that they should. Another point mutation is insertion. So this is where an A, T, C, or G is shoved into the order of the DNA. What this means is all of the rest of the DNA from that point on has to move down to make room for that base. That is going to change the order of the RNA, and it really does have a pretty big impact on the protein order. So the amino acids are then shifted and changed, and that completely changes the protein. An example I have for you is called Fragile X syndrome, and this can lead to essentially the X chromosome being irregular, mutated essentially, and it can lead to physical deformities as well as potentially learning disabilities in individuals. For nucleotide deletion, this is kind of the opposite but very similar of insertion. Instead of shoving a base into the order of the DNA, you remove a base and it causes the rest of the chain to move down to fill the space. So what this is going to cause is the RNA sequence to be changed, which is going to change the amino acids and thus change the protein. An example of something that we've actually already talked about that is nucleotide deletion is cystic fibrosis. So that is that condition that causes the plasma membrane on the cells that line the respiratory tract to produce this thick, sticky mucus. And that's the kind of why. That's the type of mutation it is. It is nucleotide deletion. All right, and we are on to chromosomal aberrations. This is going to be where not a base is changed, but an entire gene is affected in some capacity. So the first chromosomal aberration we have is gene deletion. So we have our normal chromosome and our genes on the top of the illustration. We are on the bottom left, where we see gene 2 has been deleted. It's just completely gone. It's not there. A condition in humans that is affected by this type of mutation is Cry-de-Chat syndrome, and it is a condition that has quite a few different symptoms, potentially an irregular shaped head, maybe a cleft palate, a handful of other things, but the most notable, and the reason it is called Cry-de-Chat, which is French for cry of the cat, is that the infant newborn will cry in this yowling like fashion that is very similar to like a cat crying. So that is one of the most notable features of this condition, aside from other physical malformities. The next mutation is gene relocation. So in this example, we are taking gene 1 and we are moving it to a completely different chromosome. The example I have for you is actually not a human one, but it does affect humans. This is a bacteria that obtained gene relocation of genes from a fungus. So a fungi chromosome jumped from its DNA into the DNA of this bacteria called Legionella. And if you contract an infection of Legionella because of that gene, it's a particularly tricky bacteria to get rid of because of that gene. It makes it a little bit more potent, as well as a little bit more dangerous, and it has kind of essentially bacterial superpowers that it inherited from a fungus. The last chromosomal aberration is gene duplication. So we see there that gene 3 has been duplicated. There are 2 gene 3s. This can be an issue because we don't want multiple genes of certain genes. So what this causes in humans, as far as an example goes, is called Charcot-Marie tooth disease. Now, this is a person's name or multiple people's name. It has nothing to do with your teeth, but it is the duplication of a particular gene which results in nerve damage and muscular weakness. And one of the kind of more physically noticeable deformities is within the foot, so a very high arch, as well as hammer toes. And I believe the only way to really try to remedy that is through surgery, and that's not a guarantee that it's actually going to help that deformity, unfortunately. Okie dokie. So, what causes all these different mutations to even occur? Well, the first kind of primary cause is spontaneous mutations. So, spontaneous meaning they kind of just poof into existence, but it's technically the process of DNA replication when a cell is making another cell, because remember, you have to have a pre-existing cell to make a new cell. So when our cells make new cells, because your skin cells make more skin cells, your liver cells make more liver cells, we have to first replicate the DNA in the nucleus. And by doing that, sometimes a mistake can occur in that replication process. So I want you to think of it like this. If I asked you, get out a piece of paper right now, and I want you to copy all the notes that you just took, word for word, perfectly, do you think you could do it? Most of you are probably saying no, and that's right. So your cells do make mistakes when they copy your DNA, and that can lead to these mutations, and it has. There are also environmental factors that can lead to mutations. These are going to mostly be mutations that are going to be in non-sex cells, whereas spontaneous mutations could be in sex cells or non-sex cells. So we have two types of environmental factors that can cause a mutation, like radiation-induced mutations. This would be like a sunburn. So the exposure to radiation from the sun causes your skin to produce more melanin. So melanin is the pigment in your skin. That pigment is meant to protect you. Now, as that exposure to the sun increases the melanin in your skin, it's trying to keep up. It's kind of like an arms race, which is why sunscreen is so important, because it adds a protective layer against that radiation from the sun. So if you have a burn, that means you have damaged your cells to the point that they did not keep up producing that melanin. If they did, you just have like a nice tan. So if you do have a burn and you frequently tan or burn and you don't use sunscreen, these cells are becoming damaged in such a way that they're constantly kind of damaged and replace themselves, damaged and replace themselves. They're trying to keep up with the damage that you're producing. This unfortunately can lead to mutations in those cells and thus cause something like skin cancer. Chemical-induced mutations, I want you to think of like smoking cigarettes. Now, the jury's still kind of out when it comes to the damage that vaping or e-cigarettes can cause, because yes, you are reducing the number of chemicals that you are inhaling, but it's not safe. Safe is not doing that. Not inhaling something that is going into your lungs, where you have very sensitive cells and tissues that are being exposed to those chemicals. So let's go back to smoking cigarettes. Cigarettes will contain some like 300 to 400 different chemicals, and a lot of those are carcinogens or cancer-causing agents. By inhaling those chemicals into your lungs, you are exposing those cells and tissues to the carcinogens, which can mutate the DNA in the cells that line your respiratory tract, as well as pretty much every cell in your body, because by smoking cigarettes, you increase your odds of every single type of cancer. So, my little PSA is wear sunscreen, And don't smoke cigarettes. And if you can avoid it, don't smoke at all. So yeah, those are some of those chief causes of how we get those different kinds of mutations. And with that, friends, that is the end of chapter 6. If you have absolutely any questions at all, please don't hesitate to reach out or email me.